Good morning, everybody. We have here today with us Professor Ilya Shmulevich from the Institute for Systems Biology in Seattle. Ilya received his PhD in ECE at Purdue University, working on music signal processing, after which he was a researcher at Tampere University in Finland, working on nonlinear systems theory, statistical signal processing, computational learning theory, and so forth. He's very well known for co-developing, along with Ed Doherty and Wei Zhang, probabilistic Boolean networks, which he's written a famous book on, which are models for gene regulatory networks. Since 2005, he's been on the faculty at the Institute for Systems Biology, where he has been pioneering systems approaches to biological systems, in particular focusing on multi-scale modeling of cancer. So without further ado, let's welcome Ilya to talk about information-theoretic perspectives on stability-responsiveness trade-offs in biological systems.
Thank you very much.
Ilya.
Thank you. Hello, everyone. Thank you very much for the introduction, Sriram. I'm very glad to be here. Also, thank you for inviting me. I'm a double E, as Sriram said, so I'm very glad to be here sort of in my home department. I'm also very proud to be an affiliate professor here in the double E department, and I hope to establish more collaborative ties with the double E department. And the topic of my talk today is on what can information theory or information-theoretic approaches tell us about trade-offs that biological systems must necessarily make. So as engineers, I will just hover on this title slide for just a moment, just to kind of set up the motivation. As engineers, I think we're very used to thinking of trade-offs, and we have good ways of measuring stability of systems because we don't want design systems to blow up in one way or another. And presumably, biological systems also need to do that. They need to somehow be stable under a variable environment. But at the same time, we also expect many engineered systems, as well as certainly biological systems, to be responsive to changes in the environment in order to be adaptive. And so it is this motivation that I wish to discuss today. How do biological systems make that trade-off? And how can we quantify it? How can we think about it? Okay, let me see if I can move this. Okay, so a little bit of background of how we can think of cells. We can think of cells as complex dynamical systems that are composed of many interacting biomolecules, be it nucleic acids, or proteins, or lipids, or metabolites, and so on and so forth. So these are complex dynamical systems that essentially govern the cell's behavior, and the cell is the emergent property of that system. And the same, of course, is true at multiple scales of biological organization. So an organism is a dynamical system composed of many interacting cells, which themselves are dynamical systems. So, for example, the brain is a dynamical system of interacting neurons, and the immune system is a system of interacting immune cells that interact via diffusible molecules, and so on. So in biology, everything is pretty much a complex system, right? So, and coming back to what happens inside the cell, inside, let's say, the nucleus of a cell, a genetic network, a network of genes and their products, the proteins, and other molecules, essentially govern the cell's behavior and how a cell switches states, how it switches between, for example, being in a state of division to a cell of quiescence, or a cell or a state of death, programmed cell death, or apoptosis. So all of those behaviors are controlled by these internal networks. So in a living cell, these networks not only govern the behavior, but they also propagate information from outside the cell. So this is an example of a signaling network in the macrophage, which is an important immune cell type that is resident in all of our tissues and is sort of our first line of defense against invaders like bacteria and viruses, other than our skin, which is really our first line of defense. But these molecules here propagate information from outside the cell down to the nucleus through chemical reactions like phosphorylations and other chemical modifications. So we can sort of think of information flowing through these networks. And in many cases, it doesn't really matter what the actual mechanisms are. What really matters is what the interactions are, what is the overall structure of the system, and not so much how the connections are implemented. So for instance, here you see examples of three very different types of systems that exhibit very similar overall emergent behavior. The first is a soil-dwelling bacterium. The second one is the famous Belousov-Zhabotinsky chemical oscillator waves. And the third one is wave movements in the slime mold Dictyostelium discodium. So very similar types of behaviors, these spiral waves, can occur in very different systems because it's the interactions that really matter and not so much the constituents of the system. So the same, of course, is true for many open systems. Biological systems are open systems, and just like our planet, our ecosystem, it's an open system. We get energy in the form of solar radiation, and then that radiation is emitted back in the form of long wavelength radiation back into the atmosphere. So you have influx, you have outflux, but on average, the temperature more or less stays the same. Right? So this is an open system, a thermodynamically open system, just like a cell. And this system must be able to adapt to changes. Just like a planet is changing and the climate may be changing, or is changing, the same thing with a cell. A cell constantly needs to be changing and adapt to a changing environment. What I mean by that is that environment may have differences in light, in the chemical constituents of the environment, and so on. And so the cell needs to be able to adapt to that, and yet maintain stability. So therein lies this fundamental trade-off. It needs to be stable in that phase of a changing environment in order to do whatever the cell needs to do to continue doing it. But it also needs to be adaptable. It needs to adapt to these changes when they do occur. So it needs to be malleable, but it cannot be too malleable because then it will be too fragile. Because if the environment is changing too rapidly or too chaotically, then if the cell is just trying to keep up with the environment, it will lose its stability. So sort of intuitively, you can feel that the cell needs to strike this trade-off. Okay. So this leads to this idea of systems that are poised at a phase transition between an ordered regime and a disordered regime. And these are called systems that are critical systems or poised at criticality. One way to think of such systems is that they are essentially dynamical fractals. If you look at the dynamics, they are fractal, essentially. So that means, and I don't want to belabor this point about why these kind of scale-free laws, why they're fractal, because they often exhibit power laws. But you do see these kinds of behaviors for example, in the famous Ising model that many people in engineering and image processing and other fields and physics use. But in many other systems that fall within the same universality classes as these systems, you see these kinds of power law behaviors, where essentially the system, the correlations start to acquire a power law distribution as a function of space, for example. So the correlations between atoms in a magnet die out as a power law as a function of the distance between them, only when the system is critical. So it loses a characteristic scale. It becomes scale-free, just like these power law distributions. And you see this in many natural systems, not just in biological systems, but in even geological systems, like sedimentary systems. So this is a classic example of two rocks, upper and lower one of two rocks. And these are, oops, sorry, that was a giveaway. So these are sort of markers, because usually you need to show a marker like a coin to show how big that rock is and how big that structure is. So here's one. And then if you actually see that this one is a finger pointing to this marker, and here is a person. I don't know how well you can see that, but there's a person standing next to it. So this is a big face of a big cliff, whereas this is a small rock, but visually they pretty much look the same. It's kind of scale-free, which suggests that the processes that led to the formation of these structures were somehow scale-free as well. Okay, so the tool that we would like to use to study biological systems at first is a model system that is called Boolean networks, which was introduced by Stuart Kauffman, who was also the person that has stated this really intriguing hypothesis that some people refer to as life at the edge of chaos, that living systems are indeed poised at this critical phase transition between an ordered and a disordered regime. And in the past several decades, there have been many papers, probably now in the hundreds, in the areas of neuroscience and insect colonies and bacterial colonies and many other kinds of systems, including gene expression and cells, that have shown evidence that biological systems are poised at this critical phase transition. I would like to tell you about some of that now, but also motivated from the perspective of information theory. But first, let me introduce one of the tools that we will use, and these are Boolean networks. So a Boolean network is a model system that represents relationships between genes. Genes are idealized to be on-off devices, basically bits, zeros and ones, and they're wired together using, what they call a wiring diagram. But importantly, they are interconnected with Boolean functions that govern the behavior of these genes as a function of some other input genes to which they are wired in the wiring diagram. So that is to say, sorry for the fuzziness here, that the activity of the ith gene at time t+1 is governed by some Boolean function assigned to gene I as a function of some other genes at time t, at the previous time point, determined by the wiring diagram. The connectivity is an important parameter in such a network. How interconnected is the network? K is the average connectivity, okay? You can have K be constant, where every gene has the same number of input genes into its Boolean function. Or each gene can have a different number of inputs, Ki, and then capital K is just the mean number of input variables. And it can be chosen from different distributions. Another important parameter in a Boolean network is the bias. When you generate a random Boolean network, you typically generate a random truth table. So if you have Ki inputs, you need to generate 2 to the Ki entries in the truth table. And typically you flip a coin to generate a random truth table, and you flip a coin with probability p. That's the bias. So if it's unbiased, you'll roughly have the same number of zeros and ones in the truth table. And if it's biased, then you can have more ones or more zeros. That's an important parameter for generating random Boolean networks. It turns out, and this is a result that goes back to 1986, to Derrida and Pamot, and this is this famous critical transition curve that you see here. In terms of these two parameters, K, which is the average connectivity, and P, which is the bias, you can determine whether this dynamical system, the Boolean network, will be in the ordered or disordered regime, or whether it will be in the critical regime. And this is the critical transition curve, shown by this curve. So if you are above this curve, where it's equal to 1, you have chaotic behavior, and I'll explain what that means. If you're below the curve, you have ordered behavior. And if you're exactly on the curve, you're right between order and chaos. And that's the critical phase transition curve. It also turns out to be the Lyapunov exponent, the same one that we know and love for continuous dynamical systems. This is the Lyapunov exponent for discrete dynamical systems, or Boolean networks in this case. So what that means, for what chaos and order means, is that nearby trajectories either converge or diverge. So imagine in the limit when you have many genes, when n goes to infinity in the thermodynamic limit. Imagine that you take two identical states, except that one of them you perturb by 1 bit. You only flip 1 bit, so you make the Hamming distance 1 between those two states. So it's a minimal perturbation, sort of like an epsilon perturbation in a continuous dynamical system. And then you ask the question, what happens to the successor states? Do the successor states get closer, or do they get further apart? And then you do that many times on average to get average behavior, and you can essentially get an estimate of the Lyapunov exponent. You can see whether the system tends to converge or diverge in its state space. And this is exactly what happens in the thermodynamic limit. And you can prove this using the mean field approximation. Do you have a question?
Yeah, so just above the line, we would see divergence a little more than 50% of the time, and we call that chaotic?
Yes. Above the line, you will see divergence. I don't know if it would be more than 50% of the time, but the higher above the curve you go, the more you'll see it. So if you're close to the curve, you won't see it very much. But then the further up you go, the more chaotic it will get. Yes?
So on the curve, how often will you see divergence?
On the curve, the question is what happens when you're on the curve. So on the curve, you will see both types of behavior. You see a very special type of behavior on the curve. Sometimes you will see convergence, sometimes you will see divergence, but on average, you will see neither. So on average, the Lyapunov exponent here will be 0. And I'll show you how to define that. So it's a very special regime. It's a very narrow regime. Just like in the Ising model, it's a very, very narrow regime when you go through this critical temperature. So in the Boolean world, we can define the Lyapunov exponent as follows. We can start by defining the partial derivative of the Boolean function. And yes, such a thing actually exists, partial derivatives of Boolean functions, and they are defined in a very similar way to regular derivatives. And that is that we simply stick a 0 or a 1 in the ith variable, and then we ask whether the function changes. So we take exclusive OR of the Boolean function when you stick a 0 or a 1 into that input, okay? That's all that means. So it's addition modulo 2 of these f0 and f1, where f0 and f1 just represents sticking a 0 or a 1 in the ith variable. Okay, that's the partial derivative. So the partial derivative is a Boolean function, right? This is a bona fide Boolean function, but minus 1 variable. It doesn't have the ith variable, because now I've stuck a 0 or 1 into it, right? Now, what I do is I take that Boolean function and I take the average of it. I take the norm of that Boolean function, where the norm is defined just like this. I add up the values of the function over all of the inputs, basically go through the hypercube, add it up, and divide by 2 to the n, just to get it between 0 and 1. That's the average. So the activity of the ith variable of the function is defined as the average of the partial derivative. So in other words, on average, how often does the function change if I change the ith input? That's what it means. Okay. Now, we're ready to define the Lyapunov exponent. The Lyapunov exponent, also known as the sensitivity in this Boolean world, is simply, well, first let me define the sensitivity and then you'll see the punchline. The sensitivity is defined as follows. It is all of the sensitivity on a vector x of the function f is all of the Hamming neighbors of vector x. So here's what that means. X addition the unit vector EI. That means take vector x and go to all of the Hamming neighbors, to basically change one bit at a time and look at all the Hamming neighbors on the cube for vector x and ask the question, on how many of those neighbors is the function going to be different from x? And that's called the sensitivity. Okay? And that can be defined simply as the sum of these indicator functions of the number of times that f is not equal, f of x on the Hamming neighbors of x is not equal to f of x. And now if we take the average of the sensitivity, this is just vector x. Now I want to do it over all vectors x, over the whole cube. So I want to take the expectation of this sensitivity, the expected sensitivity. So I put an expectation inside here. It goes inside the summation. And expectation of this indicator function, since it's binary, is just the probability. It's just the probability that this f of the Hamming neighbor is not equal to f of x. The Hamming neighbors of x is not equal to f of x. But that's simply the activity. On the previous slide, I already defined that. This is exactly this probability. To see that, you will simply see that the expectation of this indicator function is the probability that the function on its Hamming neighbor is different. So the average sensitivity becomes the sum of these activities. So this gives me an analytic tool to compute the Lyapunov exponent of a Boolean network. If you give me a Boolean network, I can tell you precisely what the Lyapunov exponent is just by following these calculations of activities and average sensitivities of the Boolean functions. Okay, since we know that, we know precisely how to tune these networks to undergo phase transitions. We can tune K, we can tune P, we can tune other things that I won't talk about because that's not important. The point is that we have tools to create large model networks that can be with any Lyapunov exponent. They can be ordered, they can be chaotic, or they can be precisely on the critical phase transition. Okay, now, over the years, over the past 15 or so years, there has been accumulating evidence that cells also operate in this critical regime, as I have already mentioned. A lot of evidence has accumulated in neuroscience, measuring brain oscillations using EEG and MEG and functional MRI. There are very interesting papers showing criticality in the brain. But in cells, it's more difficult to get good data, especially time series data. And so there has been some attempts in the literature to show that cells are indeed poised in this critical phase transition. And there has been evidence, it's been accumulating. And what I'd like to talk to you about is an approach that's based on information theory. So the way we can think about Lyapunov exponents, we can think of it in the same way, but in terms of information. So for instance, imagine that you have two states of a system, and they are somehow slightly different by epsilon or some difference. Then you can again ask the question, well, what happens on the successor states? Do they converge or do they diverge? Except now the question is posed in terms of informational distance. We can ask what happens to informational distances between these states. Do they informationally converge or informationally diverge? And what does it even mean to even say that? And before I show you how we address this problem, I would like to make the point that in either case, information would be lost. Because here you have a system that is in two different states. And imagine that the successor states are converging. If they're converging, that means the system sort of forgets what it discriminated before. If a biological system needs to take actions based on discriminations, it has lost that ability if it converges too fast, because two things that were different are no longer different in the future. You have convergence. Okay, so you lost information. Now imagine the opposite situation where you have divergence. Here you have two states which are pretty similar, and maybe those are just fluctuations, and the system really shouldn't be discriminating between them. And now the system is diverging, and you're going off in two different parts of the state space. So again, you're losing information. So in either case, the system is unable to coordinate its behavior, and it's losing information in the sense of tying past discriminations to future actions. So this is akin to Lyapunov exponent, but sort of in an informational sense. And the tool that we have used to measure this is algorithmic information theory. It's Kolmogorov complexity, which is, in a sense, the absolute measure of information in an object, in a string, x, which can be represented as the length of the shortest binary program to output x on a universal Turing machine. How much time do I have, actually? 11:30. Okay, good to know. So, on a universal computer or on a universal Turing machine. So it's kind of a funky definition, but it's also very simple. Unfortunately, Kolmogorov complexity is not computable because we can never find the shortest computer program or the shortest universal Turing machine to output a given string x. But nonetheless, that's how it's defined, and in terms of information, it can be thought of as sort of the ultimate lossless compressor. It's the amount of information embedded. If you could compress it without losslessly in the most ultimate way, you can't compress it any further. That would be the amount of information in that object, in x. Right? And the program k would generate x, and the Kolmogorov complexity would just be the size of that program. So this really is different from probability theory in the sense that it discriminates between cases that the probability theory does not. If you consider coin flips, and if I showed you a sequence of coin flips like this, and I asked you, well, do you believe me that I flipped those coins this way and this was the outcome? You might want to count how many heads or tails, or maybe do some other statistical tests and get some p-values and maybe say, yes, sure, I believe you. But then if I showed you something like this, it might pass some of your statistical tests, but you probably won't believe me because it's too regular. And you'll say, well, wait a minute, no way you flipped a coin that many times and you got heads and tails. You know, it was such a regular pattern. And of course, you could devise a statistical test to say why that's not possible either. If you look at sort of joint probabilities, or well, not joint because they're independent, of course, but you could always pose something in the language of a statistical test. But in the language of Kolmogorov complexity or the algorithmic information theory, this is answered very, very simply. It simply, intuitively says, well, how big is the program to generate that upper string? Imagine that was just like 1,000,000 digits, right? Well, if it's really, really, really random, the shortest program might be print string. You can't really compress it too much, right? Maybe a little. So it'll be roughly the same size as the string. But this one, if I had 1,000,000 digits of heads and tails, the program would be tiny. I'll just say I'll have a for loop with print heads and tails, and it's good. So this has a very small Kolmogorov complexity because it's very compressible in a sense. Even though the probability of that sequence is the same as probability of that sequence in terms of just probabilities of flipping coins. Okay. So using this idea, we can define an information distance. Okay, now this is some really beautiful work that came out in information theory in the 90s, where people have defined information distance using these algorithmic information theory tools that is similar to Shannon's mutual information, but it does not require probability distributions. It can be basically applied to any objects that is represented by a string, or it can be stored on a computer. So it can be a network. It could be a genome sequence. It could be whatever string you want it to be. And this information distance could be used to quantify how similar the two different objects are without any kind of parameters. And the way it's defined, or the way it can be implemented, because Kolmogorov complexity is not computable, the way it can be implemented in the real world is actually using compressors, lossless compressors like bzip2 or gzip or other compressors. And the idea is to take two strings, x and y, compress them separately, and then also compress them together. So basically, how well do they compress together? Typically, people just concatenate them. And then you look at the difference between the compressibility together and separately using this formula, and you get a distance called the normalized compression distance. Okay, I should have put references on the slide. There's some beautiful papers from Vitani and Chilibrasi in information theory transactions. So using this kind of a distance, we can look at dynamical systems. Because imagine now that x and y are just states. Those are states of a dynamical system. Now I can represent informational distance between two states, and I can ask what happens and there's successor states, just like a Lyapunov exponent. I can use this information distance as a tool. Well, here's one curve for Boolean networks. Now, the reason I wanted to introduce Boolean networks to you is because we have all the tools we need for Boolean networks to talk about criticality and phase transitions, because they've been studied a lot, and we have all of the tools. But now we wanted to see whether this approach based on normalized compression distance, this informational approach, would even work for Boolean networks where we actually know what to expect. So this is the so-called Derrida curve. What this does is it measures the distance between two states at time t on the x-axis and the distance between the successor states at time t+1 on the y-axis. Okay? So if all of your points essentially fall on the diagonal, that means that you are neither converging nor diverging. If your points are above the diagonal, that means that you are on average diverging. You're going into the chaotic regime. And if you're below the diagonal, you're on average converging, and you're in the ordered regime. And so this is a kind of a way to look at the states, at this phase transitions. And for Boolean networks, remember we had this phase transition curve. Yes, sir?
Sorry, I'm just missing one thing.
Yes.
I thought the MCD had two arguments, x and y.
Yes, and those are right. So
What's the t and t+1?
that is the distance, that is the distance between two states at time t. So the two states would be x and y at time t, and you plot that on the x-axis. And then you look at their successor states, and you do it again. You plot it on the y-axis. And you do that many, many, many times, you get a curve. And that's the curve you get.
So it's not the distance from t to t+1 on one state.
Right, exactly. It's not the distance from t to t+1 on one state. It's always between a pair of states. And so here we get exactly the behavior that we expect. The red curve, I'm sorry, I'm pressing the wrong button all the time. The red curve is exactly on the diagonal, and that is exactly corresponds to critical Boolean networks when k equals 2. If you remember that critical phase transition curve, 2kp, 1 minus p equals 1, the one where it had chaos above and order below, that curve, that's exactly what we use here to know whether we expect ordered behavior or chaotic behavior. And sure enough, this information distance exactly recapitulates what we already know. Okay, then we can also try other Boolean networks, like scale-free Boolean networks. Those are Boolean networks where the wiring diagram has a scale-free distribution as opposed to a constant distribution. And here, there are also some theory. There was another paper published a while ago where they worked out all the theory for scale-free networks for phase transitions. Sure enough, make a long story short, information distance completely correctly captures this red curve as the critical curve, and it corresponds to critical scale-free networks. Even ternary networks, okay, forget Boolean networks. Now genes have 3 values. It's not just 0 and 1, they have 3 values now. It turns out that there's also a theory has been worked out for that, for any k-ary networks, not just ternary, but any number of levels. And it turns out that for ternary networks, the average connectivity is 1.5, which is the black curve, exactly falls on the diagonal. Right, so even for ternary networks, the information distance works correctly. So this is nice, because the compressor doesn't really care if it's compressing binary data or ternary data or what kind of data it's compressing. It's just compressing stuff, right? And it correctly captures the dynamical behavior here. So then we tried applying this to macrophage data. So this is this immune cell type that I mentioned. And we actually had time series data after stimulating the cell with different ligands, or so different bacterial pathogen-associated ligands. And measuring many different states at different time points, we were able to construct an empirical version of this Derrida curve. So take 2 states, measure the information distance, put it on the x-axis. Now look at their successor states, measure the information distance, put it on the y-axis, plop a point on the, put a point on the clock. So every one of those points, it was a pair of states followed by a pair of successor states. And there was evidence, this is, again, somewhat weak evidence, but there was evidence that all of these points fall on the diagonal. No matter how you discretize the data, binary discretization, ternary discretization. But here's was the cool part. Then we took some data that was from a knockout mouse, where the macrophages in this knockout mouse were very, very hypersensitive. So essentially, if you challenge these macrophages with some ligand, they will react in a hypersensitive way. And usually it kills the animal, in fact, when they're challenged by infection. So when we looked at the data gathered from those mice, we saw, this is the ATF3 knockout mice, we saw that there seems to be evidence that all of the points lie above the diagonal. So informational distances kind of diverge. They spread apart, suggesting that perhaps this is an evidence of chaotic behavior, hypersensitive immune response. But this is very circumstantial evidence. So, I mean, so to speak, this is not very strong. So, now I want to just say a few words about also information storage and information diversity of these complex systems. Why would a system want to be in the critical regime, right? I mean, we already sort of discussed this idea of tying past discrimination to future actions and not losing information. But there are other benefits, so to speak, of being critical. One of these benefits is information storage. Imagine that a complex system can be used to store information in the sense of associative memory, just like a neural network. So if you start with certain states, you will end up in certain attractor states. And if you start with certain other states, you might end up with certain other attractor states. So that is a type of associative memory. The system essentially associates a bunch of states in its state space with an attractor, if you think of an attractor as a label, to which the system will flow, having started in any one of those states. So the state space of the dynamical system is partitioned into basins of attraction. All the states in one basin of attraction will necessarily flow towards that attractor, and all the states in a different basin of attraction will flow to their respective attractor. Okay, so it's a partitioning of the state space. So then you can ask, how is that state space partitioned? What is the partitioning? Can we quantify this partitioning? And so we can define the entropy of this partitioning, the entropy of this, which we call the basin entropy. How does the state space get partitioned by the dynamics of the system? For example, if all of the states fall into one attractor, that's not a very good partition, right? It's a very, very hyperstable attractor, and that's it. But if every state is its own attractor, that's also not very good. because it's not stable at all, right? It has no stability. And so you can sort of measure this. You can measure this Bayesian entropy. It's a type of information storage, a type of memory of the dynamical system. Okay, so long story short, what happens? It turns out that for critical networks, if you run this for many, many types of networks and you tune them to be critical or ordered or chaotic, the information storage, or the Bayesian entropy, is maximized. And this is provably so. In the thermodynamic limit, you can actually prove that the Bayesian entropy will scale with system size only in the critical regime. So if I make my system larger, I can make my memory larger. But only if I'm critical. If I'm chaotic or I'm ordered, I don't care how large my system gets, I cannot increase its memory this way. Okay, so that's provable. Here's another interesting result. Critical systems are also diverse. They exhibit a type of diversity that can be very useful in an evolutionary sense. So when we looked at Boolean networks in different regimes, for example, chaotic regimes, ordered regimes, critical regimes, and we even looked at Boolean networks that had different types of wiring, not random wiring, but not only random wiring, but also regular wiring, like on a cellular automaton, like on a grid. And then we looked at the normalized compression distance between the structure of two networks and between the dynamics of two networks. This is another beautiful thing about this information distance. You can give it anything you want. So we gave it two structures, which is to say two wiring diagrams and sets of Boolean functions that define the structure of my network. So that's my x and y. And then we use those networks to generate a bunch of dynamics, a bunch of states. We generate the state trajectories, and now that's my x and y, and I can look at the information distance between their dynamics. So I can look at the information distance between their structure and between their dynamics at the same time. And I can put one on the x-axis and one on the y-axis. And what we found is that critical networks, shown over here, maximize the diversity of their dynamics relative to their structure. So that means with a similar type of structure, you can get very, very different dynamics if you're critical. If you're not critical, then your structure very much constrains the types of dynamics you can have. Whether you're ordered or you're chaotic, you're very much constrained. But if you're critical, you can have very different types of dynamics. Okay, so there's a type of diversity that it affords. And very quickly, there's another measure called set complexity, which we introduced, and we showed that the set complexity is also maximized at the critical regime, so I won't dwell on this. So the nice thing about this informational measure is that it can be applied to anything. Of course, therein also lies its weakness. But the nice thing about it, that sort of operationally, is that you can imagine running a computational model of a biological system that's essentially like a computer program that is modeling the biological system. And then, if you can represent the state of that model using some string on a computer, then you can use this information distance to analyze its dynamics. Okay? So as a proof of principle, we took a simple published model, it's called an executable model, because it's a model of cells interacting with each other. It's sort of a toy model, but it's an important proof of principle for this information distance. Because this model works on a grid. The cells can move on a grid. They can move left or right or up and down. They can also interact with each other by secreting molecules, by secreting diffusible molecules, and they have receptors that they can read the molecules that have been secreted from other cells. And the cells themselves can go into different states. So all of that is represented by a snapshot, a state of the cell, or of the system, excuse me. The grid location, what cell is in the grid location? What are the secreted molecules? Is it moving? Is it proliferating? Is it dying? What's the movement type? This is very non-quantitative data. This is kind of very messy type of information, right? So normally, how would you analyze the dynamics of this sort of system, right? You can't use tools from, dynamical systems theory, because these are very, heterogeneous kinds of systems, executable models. So with this information distance, we can simply take this state and apply the information distance or normalized compression distance to two states and compute their distance. Then look at their successor states and compute their distance again and get the same type of Derrida curve. Okay, so moreover, we can make perturbations in the model. We can knock out some genes in the model, and then we can see what happens. So these are just trajectories projected with multidimensional scaling to make it look nice. But this shows that the state trajectories are different when you knock out different genes. Okay, so that's good. But here are these Derrida curves. So just like before, the x-axis is the distance between two states at time t. The y-axis is the distance between two states at time t plus 1. And the different graphs show what happens when you knock out different genes. Like this is a knockout of interferon gamma. This is a knockout of interleukin 10, and so on. And you can see that you actually start to see different behavior where different dynamical state trajectories and transitions start to accumulate in different parts of the state space, where some of them you start to see ordered behavior, some you start to see chaotic behavior in different cases. So this is just a kind of a proof of principle showing that this informational distance could be used to analyze even an executable computer model like this of a biological system. It's very general. Okay, and now in the last, I guess, 5 minutes, and I haven't left much time for this, I would like to tell you a little bit about a related approach based on time-frequency representations. And I know that I will not do justice to this topic by telling it to you in about 5 or 10 minutes. But if you are sufficiently interested in it, then please contact me by email or however you like, and then we can talk about it. But the reason I wanted to include it here is because it is also a way to quantify the trade-off between stability and responsiveness of a biological system. And it's not really directly information theory based, so it's based on time-frequency representations. And we looked at several biological systems for which we actually had mathematical models. So one of these is this small circuit of oleate acid metabolism in yeast. These are transcription factors, and they sort of form this feed-forward loop, overlapping coherent feed-forward loop. And there are ordinary differential equations, nonlinear equations that model the mRNA levels and the protein levels and the various kinetics of these molecules. Okay? And the parameters are experimentally determined, and they have been published elsewhere by our team. But now, if we look at the stability versus responsiveness in terms of such a system, we can look at it also in terms of maybe signal processing. There's also a trade-off in signal processing where we want to remove noise. That's noise suppression. But we also want to preserve details. We don't want to remove the details when we remove the noise, right? We just want to remove the unwanted noise while preserving the details. It's sort of competing requirements. So a biological system wants to do that too, right? It wants to remove the noise from the environment, but it wants to preserve important details. And indeed, there are examples of this in the literature. For example, E. coli is essentially implementing a Kalman filter, where it's swimming up a gradient of sugar, and it's implementing optimal state estimation from a noisy input. And so a cell definitely tries to kind of implement this sort of this idea of estimating something in the presence of noise, because that's what it needs to survive. So now we took another circuit which was also characterized mathematically, galactose. So this is oleate. This is a fatty acid. Yeast cells don't really like it, but they'll eat it if they have to, if there's nothing else. And this is galactose. They definitely prefer galactose, and they will eat this up much more quickly. So this circuit, which is an interlinked positive and negative feedback loop circuit, has a different architecture than this circuit, and it is believed to be, well, and actually proven to be much more responsive. And so we know that from linear systems theory, we know that the system is characterized by the impulse response. The impulse response fully characterizes the behavior of the system, but that's certainly not the case for nonlinear systems. In other words, it's very much input dependent. So in order to characterize typically a nonlinear system, excuse me please, in order to characterize a nonlinear system, we need to look at all or many possible inputs and outputs and try to use the collection of inputs, outputs to characterize some behavior of the system. And this is exactly the approach that we took, using time-frequency representations, as I mentioned, using so-called spectrograms, which are based on short-time Fourier transforms. Of course, it could be done based on other multi-resolution methods, such as wavelets or other multi-resolution approaches, but we just decided not to focus on that, to do something simple and just to use a simple short-time Fourier transform. And the idea is the following. Take your model, which is a set of differential equations, as I showed, give it a bunch of inputs, stimuli, measure a bunch of responses from those inputs, and construct spectrograms using the short-time Fourier transforms from the inputs and the outputs. Do that a bunch of times with different noise levels and different types of signals, and start gathering statistics from these spectrograms that tell you something about the system, specifically something about the responsiveness and stability of the system. Okay. So as I said, this will be a fly-through in these last 5 minutes on this topic, but one way that you can measure the stability is to look at the mean frequency. In each frequency band of the spectrogram, you can take the mean and then look at the mean frequency in the input and the output, take the ratio, and that can be a measure of stability. There can be many other measures of stability based on these spectrograms, but that's one. A measure of responsiveness one measure of responsiveness could be to look at total variation of these transform coefficients. In each frequency band over time, you look at the total variation of the coefficients, of the spectrogram coefficients, and then you look at the input and output total variations in each frequency band and determine the Kolmogorov divergence between those distributions. And the responsiveness becomes 1 over the Kolmogorov divergence, which is to say that if they're very, very similar, it's not very responsive. Excuse me, it is very responsive. If the divergence is 0, then this will be very, very responsive because it's 1 over 0. Okay, so there are, of course, many other approaches, but we just took those two approaches as a proof of principle. Then we started looking at the galactose and the oleate circuits, and sure enough, when we started plotting the stability versus responsiveness, we found very, very different behaviors. And when you actually you can do a knockout of the model, again, knockout, in the equation, knock out a gene and see what happens, you can actually take the oleate model and make it look like the galactose model in terms of its stability, responsiveness, behavior. And then finally, this is kind of the most interesting part, and again, I apologize for rushing this very last part. These are what we call portraits of stability and responsiveness. You can take the network and then take two parameters of the network, maybe the strength of some feedback loop or some feed-forward loop, and take those parameters and start tweaking them, start tuning them. And for each set of parameters, compute the stability and compute the responsiveness, doing this spectrogram ensemble approach with many signals. And then you can see how does the stability and responsiveness change as a function of your parameters of the network. And here's the wild type in the white crosshairs right here. The wild type sits right here. So if you deviate from the wild type, you start to move away from the wild type conditions. You can see that the stability rapidly drops. In the Oleate network, it seems that the system is poised at this edge of stability, almost like a Pareto optimal front of stability. And as soon as you move away from it, you lose stability. And you also become hyper-responsive. However, in the galactose network, it's not stable at all. It's highly, highly responsive in the wild type. And if you move away from it, nothing much happens. In other words, here, if you move down in this parameter space, you don't sacrifice responsiveness. So again, this is a way to look at responsiveness and stability trade-offs using time-frequency representations when you have a mathematical representation of your model, as we did here. When you don't have a mathematical representation, then you can fall back on approaches such as the information distance and the normalized compression distance, which you can just apply to data or to even executable models, as I showed. Well, with that, I'd like to thank you for your attention, and I'm happy to answer any questions in the remaining time. Thank you. Thank you.
How do you use this information? Let's say you had a model of a living system, a cell. Would you use it to evaluate health of the system? Like if the optimal state is criticality?
Yes, it's a very interesting hypothesis that if a healthy cell is indeed poised in a critical regime, could it be that an unhealthy cell or unhealthy system is somehow deviating from criticality? And some people are indeed looking at that hypothesis, for example, in the context of epilepsy, where people who are experiencing epileptic seizures may be that essentially the dynamical system is deviating from the critical or subcritical regime and kind of venturing into the chaotic regime. And yeah, so that's a very intriguing hypothesis. Yeah. You had a question there.
Can you go back to the information distance curve? So my question is that the plot, is this the expected value of distance at time?
Yes.
Or it's a deterministic?
Yeah, so the question was, is this the expected value? Do these curves represent the expected value? And the answer is yes, they do. Yeah, so these are the smoothed versions of many, many, many points.
Also, there is a distribution for distance at t plus 1 given t, right? Given distance at time t.
Indeed.
So my question is that, do we know how these distributions actually look like? Because I mean, for the systems that are ordered or chaotic, do we know how likely it is to get some distance, say, larger than the distance at time t? Is this unlikely totally for an ordered system, or we get some larger distances, but we don't?
Right, so the question is, do we have information on the actual distributions of these distances at time t and t plus 1, not just the averages? And the answer is, of course, yes, because we generate the simulations and we can save all the data, and so we can definitely look at them. We have not done so in this published paper, but the information is there to be looked at, so that would be indeed an interesting thing to explore further. Hi, Herb.
So I kind of came in late by accident, but so the two networks you had, one was fast responding, the other was not as fast responding. Do you know why, though? I mean, you can do this analysis and show, I mean, the beauty of linear systems is it'll also explain why. But can you explain why one was more responsive than the other?
Well, yeah, so the question... Yeah, so the question is, why is one system more responsive than the other system? And of course, it depends on how one interprets the question why. But you could write out the equations, the nonlinear equations, albeit, for the system and say, well, this is why. Because we can then generate some distributions, get flow cytometry data, and sure enough, we have a broader distribution, and therefore it's more responsive. So you can sort of say, well, we can answer the question why that way. Not that way. Okay.
I mean, to have some intuition, you know, why? Because if you have some intuition, you can start understanding the design principles behind these motifs, and then perhaps design new ones, investigate existing.
Yes, indeed, and you're right. And I guess we don't have that intuition. And some people are working on these motifs, like thinking, well, this motif is a smoother, and this motif is an amplifier. And I think some people are looking at it that way. And I think perhaps these tools could be useful to dig deeper into that kind of an inquiry. Yeah. Yes.
It seems like in many parts you're kind of implicitly relying on Tocken's theorem to obtain the dynamic response of things. Is that?
I don't think so. It's venturing outside my sphere of expertise, but I think Tocken's theorem is for deterministic systems.
Yeah, so I was wondering, because you're making these measurements, and I haven't really thought my question all the way through yet, but the idea is you've got something to, well, the previous questioner asked about a healthy cell being on the critical regime. So as a cell moves away from the critical regime, something needs to pull it back to criticality in order for it to heal itself in some sense. So there's a larger dynamic that's happening. So is there a sort of a Tocken-style analog that you can pull back? Can you see something about this larger dynamic as it moves away and pulls back?
Yes. So
does my question make sense at all?
I think it sort of makes sense to the extent that I understand Tocken's theorem. But I don't know, so the short answer is I don't know whether there is sort of a larger process at play that governs this drift away or towards criticality. Yeah, I mean, we sort of think of a system as an isolated system, but of course it's not. It's an open system. It gets inputs from outside, from the greater system. And so, yeah, it's very difficult to think of it that way. But I think operationally, we can certainly estimate things like the Lyapunov exponent. We can measure certain aspects of a dynamical system, and we can say, hey, you know, our estimates look like they're chaotic. Like if we measure brain oscillations, we can still kind of use numerical estimates to make some conclusions.
Right. So, okay, so I think translating back, I think the answer is that we're looking at something that's near an equilibrium, and that's basically all we're looking at, is something that's near an equilibrium. We can't really deduce anything about in equilibrium in the sense that if it pulls away from criticality, it goes back, or if it pulls away the other way. There's sort of a stable equilibria.
Okay, I see, yes. And so this actually touches on an area called self-organized criticality, SOC. And this is a field that grew out from physics in the 1980s, from Per Bach and others, and Tang and others. And the idea was, the famous metaphor was the sand pile model, where the slope of the sand pile self-organizes to a critical slope. It cannot be too high because then it'll collapse. It cannot be too low because as you start piling up the sand, the slope will go up, and it'll sort of stabilize to a certain slope that is dependent on the physics of the grains of sand. And so that was the metaphor for self-organized criticality, that also biological systems self-organize. They kind of get pulled back, as you said, to a critical regime. And so there's a whole field of inquiry about that. It's, yeah, very interesting. Okay.
Any further questions? If not, let's thank the speaker for an excellent talk.
Thank you. Thank you very much.